Okay, predict subcellular localization. We talked about these words already. So there's by homology, de novo. De novo means I don't use any information. By homology means I sort of look up similar information that is evolutionarily produced, essentially. And ab initio means I have principles. Typically, in this lecture, ab initio we're not going to touch because computational biology essentially doesn't touch ab initio. There is really no good example where we can have an entire algorithm go through. The problem is very simple. You have the cell that is highly densely packed. In this particular case, what you see is a cell. One way of looking at it is so the green bit here is the wall, and then there's sort of an inside of the cell. A bacterium essentially is like that. So all the genes, the proteins that are the machinery of life swim inside of this soup, so to speak. There are some substructures noted here as plasmids or ribosomes. Think about them as larger machineries. Okay? Those are just large groups of proteins that stick together. Stick together again means on the lifetime of a cell, they are always essentially there. They just float around and they are huge machines. But they are not in any way hindered from moving as size allows them, moving around freely in that cell. Okay? In that sense, really it's the idea you have a bag without any further constraints and proteins float through that. The constraint, again, the special thing in bacteria is the wall, and some bacteria have this flagellum, so that essentially lets them move. Upon the evolution to eukaryotes, the story got slightly more complicated. Some of bacteria got into eukaryotes, such as the mitochondria. Getting into the bacteria, they adopted specializations. Particular specialization is the nucleus here, surrounded again by a wall. In fact, by two walls. Inside of the nucleus, you have all the genetic material, or the main genetic material. So the material that makes us ourselves is essentially the one that is duplicated by duplicating the material that is in the nucleus. So here, when we talk about the human genome, then we talk about that genetic material that is inside of here. There's another place in the human cell where we have genetic material, namely the mitochondria. But most of the genetic material of the mitochondria is not essentially used to produce proteins that act in the mitochondria. That's an additional issue. The only reason why I mention the genetic material here is tiny compared to this. Tiny means it's a factor of 100 smaller. The point is that we have 100 of these mitochondria in our cells. So essentially the sum of the genetic material is almost the same. And since that is simpler, it is simpler to read. So the first attempts to study human evolution by genetic traits was through mitochondrial DNA. And the special thing about mitochondrial DNA is passed to you by your mother, to all of us by our mothers. While, as you know, the nuclear DNA contains a copy of mother and father. And again, there are other subcompartments here. So specialization is in some sense the way of eukaryotes to get a little bit more complex. Eukaryote, I mentioned that briefly on Tuesday. Karyot is the Greek word for core. The Latin word for core is nucleus. So those are the ones with a core and those are the ones without a core. And then for that compartment we use the term nucleus. In a simplified way, and most of what I talk about today sort of has this picture in mind. You essentially have a nucleus, you have some soup that swims around, and you have some extracellular space. Proteins have to get out of the cell. Why? So I say that the machinery of life essentially are proteins, but you know, if the machinery of life and the base of life is our cells, and the machinery of life are the proteins in cells, why do we need to get out? That's why this blue space is so important and we want to predict it. Now, there are different ways in which we can predict subcellular localization. We can do homology-based inference, and in a minute I will explain what I mean by that. Text similarity, motifs, and then sort of de novo prediction from sequence and structure. And we will sort of talk a little bit about all of these today. Let's begin with the homology-based inference. And there the algorithm is very simple. You have a protein, a query protein Q. You have another protein E. And for that protein E, you know the subcellular localization. And you know, when you compare the sequences of Q and E, they are somehow similar enough. Somehow similar enough means the similarity is above some threshold T. Okay? Very simple. Two proteins are sequence similar. If they are sequence similar enough, meaning above the T, then you simply copy the annotation from E to Q. Meaning, you simply say that, for instance, if that would be a protein from mouse and human, and if you know the localization for the mouse protein, then you assume that the human protein has the same localization, bless you. So the mouse protein is nuclear, the human protein is nuclear. Now, here are the two sequences, two proteins with similar sequences, what it reads in the protein alphabet, because not O, for instance, is to begin here, O is not an amino acid. Anyway, and you see there are some changes, but then there are some similarities, and looking at that, you recognize they're similar enough, so they're above T. And that brings us to this picture that I had last semester already. So if two proteins have 100% sequence identity, it's clear they have the same, essentially the same subset localization. And the further sequence identity goes down, the more you will lose the signal subcellular localization, right? And at some point you will no longer really be able to guarantee that you infer it correctly. You get into a twilight zone where there is some signal, you can make some difference between things that have a similar subcellular localization and those that don't. And then you will get into a midnight zone where essentially you don't have a sequence signal. You can no longer do that from sequence alone, because most of the things you find at that level of sequence similarity would be random. What would that level be? When do you minimally expect to hit the midnight zone? Sort of trivially in some sense. Comparing protein sequences. What would be the number? I'm going to measure similarity here by identical amino acids, by identical letters. So if you have random letters, what's the level of match you get? Roughly. Number. No, I just want to hear a number. So I give you two strings, and I ask what's the random identity, a random number of identical letters.
15%.
Why 15? Because it's a good number?
Because I think you saw something like this last semester.
No. 5% is a good answer, 15% is a good answer, now argue why. Your argument is not good enough.
Well,
I said it last year.
my argument is that there are 20 amino
Yes,
acids.
exactly. There are 20 amino acids. So, matching, if you had randomly selected 20 different letters from the alphabet, and you wrote random words with 20 amino acids, so you're randomly assigning how often you use a certain letter, and then you compare two of these randomly created strings. On average, if the amino acids were equally probable, it's 1/20, it's 5%, right? So this is one simple way of defining midnight zone. So the moment that you are going to a level where the T of the homology-based inference is 5%, you can no longer distinguish between a signal and random, right? And in fact, random is slightly higher because they're not equally distributed, and there's all kinds of things to say. But that is sort of the clear idea where roughly this end is here. As you already see here, these drawings are not quite to scale. And you are right, I did say something about 15%. I honor your memory, but it's a slightly different context. And it's sort of, that's the 15%, that's the end of the actual midnight zone for protein structure analysis. And that is sort of empirically verified zone. While now I'm sort of asking, if you knew nothing, what could you say where that roughly sits, right? Okay, but now we in fact have to find out where exactly is this threshold T that I talked about in the previous slide here, that is such that if two protein sequences have a similarity higher than that, or percentage sequence identity higher than that, then they have a similar subcellular localization. And in order to find out, what I need is a set of proteins that have similar subcellular localization. And I have to take out of that set another set, and that set should be redundancy reduced. Now, redundancy reduced means unique in terms of sequence, meaning that, say in this set here, say I had 1,000 proteins, but they essentially had all the same sequence. And it's sort of meaningless data set. So I have to throw out things that are too similar to each other, okay? And now what I'm going to do, I'm going to compare this set against that set. This set here, I redundancy reduced to avoid too much overlap. Redundancy reduced, again, it means that no pair in this set has more than a certain level of sequence identity, okay? Now, I'm telling you it's important to have a non-redundant data set. Yet, I'm comparing a non-redundant data set, and let's just put a number here. NR is defined as there is no pair of proteins AB that have a PIDE larger than 20%. So no pair in that set has more than 20% sequence identity to each other. That's essentially what that says, okay? That's one way of defining non-redundant or unique. Okay, I say it's important to have a unique set. And now I want to sort of figure out the threshold at which I can no longer do similarity, homology-based inference through sequence reading, right? And I want to sort of test where this threshold is. What is the threshold T, right? So I need to look at data. And I need to begin with a sort of sequence unique subset. But if I argue that sequence unique is important, why don't I just compare the sequence unique set against itself? Why wouldn't that give me the best number to do? If I really believe in this credo of non-redundant or unique, why don't I do unique against unique? That would be the least redundant way. What I'm showing on the whiteboard here is I'm going to go with a unique subset against the entire set. In the entire set, I said there's too much redundancy. That's why I have to create the unique set at the first place. So why don't I do unique against unique? At the risk that I'm spending the rest of the day trying to switch on and off the camera. Okay, so the questions will be recorded. I'm giving up. Any argument? It's very trivial. That would be a very good reason, and in fact that is a problem here, that this set is relatively small. But there's a much simpler reason. So again, what I want to do is I want to sort of test. Somehow I have percentage sequence identity, that's 100 to 0. And on this axis, I want to test how good is homology-based inference. So one thing that I could actually test here is precision. Precision. So of all the inferences I do at a given threshold T, so if I use the PIDE between A and B larger than T, and then I say whenever it's larger than T, I can infer the localization from one to the other. So now I'm testing different thresholds of T. What I expect is some curve. I'm just out of simplicity, call it a linear region here. What it means, it will go down. The less sequence identity I have, maybe it's not linear. What I do know is there's some value where I'm close to 100%, and there's some value where I'm close to random here. And random essentially will be depending on how many classes I have. In the case that I'm trying to show you here, subcellular localization defined as, I believe, with 3 classes, random will roughly be 33%, right? So you know this end of the story, and you roughly know this end of the story, and there's some way to connect these 2 points. And that's what I want to shake out. And I want to shake out if the way to connect these 2 points would be such that I had this one here, I could immediately choose a threshold T. Where would you put it? Where would you put the value T? Let me call this the point A, C, and B. And if you want to put a threshold point anywhere else in the geometry, you just say left or right of A, right of A or between A and B. So where do you want to put the point? So you were the first.
I would take B.
B. So can somebody argue against B? Before I let Stanislav argue against B, what you do, I believe it's not the right thing to do, but it's what almost everybody does. Okay? This is the typical choice. So now argue against it.
Well, I would probably assume A because...
No, why B?
Because B is too indecisive. Like...
What do you mean by indecisive? It's very decisive. So the way I call that, there's a clear point. What do you mean by indecisive?
I mean that I can't express right now. 1 second.
No, let's have somebody else step on it. Stanislav is on a good, in principle, on an interesting direction. follow his thread and find a different word for it, or a different argument. Essentially, you're right. You're just not saying it right. Indecisive is what people are, not an argument. Anyway, let me not grill you on that. It's very simple. It's not simple, I'm sorry. Anybody? Yeah?
Maybe the problem is that I have a relatively large region between B and A, where I essentially don't know too much.
Yes, is that speaking against B? You could say that is speaking against B,
yeah,
but this is not the argument I mean. The argument I mean is much, much simpler, essentially. What I'm saying is that if you, whatever you do, you always have some error. And let's call that error minus plus epsilon, right? If you put yourself into choosing a threshold at the steepest point of a curve, you make yourself most sensitive to this error. Because there's no other point to pick where plus minus epsilon, whatever the size of epsilon is, has more of an effect than here. That's the problem. Again, most people do this. It's a very common, I would call it, mistake. And that is the reason. You could call that indecisiveness in some sense. Maybe it's a way to translate that. But ultimately, you ought to put yourself somewhere here, or somewhere here, or somewhere. All of these points are sort of, all of them are better. Or equally, actually, here. Well, now, in principle, at least. Now, in this particular case, we want to have a point where we are certain that we are right. So this would speak for the point A. Okay? Anyway, this is just a hypothetical curve. I'm only trying to explain what we expect and what we get. Now, again, my question is, why do not use unique against unique to produce that plot? Yeah?
Because, like, as we define unique, you have the maximum similarity of like 20%, and that's like not enough to make any predictions at all.
Is everybody clear about that? Because the way I define unique, there is no pair in there that has a higher value than 20%. So meaning, say that would be the 20%. I actually, in that set alone, if I do that against itself, you are right, it's too few points, actually 0. Right? For most of this curve, you would have exactly 0 points. And that really is a small number. It's not quite what you meant, but essentially, technically you said the right thing. So we don't have, that's why we have to accept some sort of redundancy. Otherwise we can sort of not monitor the curve here in this region above. And that's what we do. So we do unique against the whole thing, and we're sort of inferring, if we had a record such as this one here from Swissprot, that the protein is a transcription factor, here is the description of what it is, the organism, the gene name, the taxonomy, so where it comes from, there's a sort of better description of function, and somewhere it says subcellular localization, nuclear, and that essentially is what we want to infer. Okay, this is what we get. I do not show you percentage sequence identity. I show you log E value of BLAST. For those of you last semester, you remember BLAST. Essentially, that's a method that finds the best superposition of two protein sequences and looks at how significant is the hit or the similarity that you have given the length or given the sequence you have at that point. The HSP distance here is, I showed you in one of the earlier slides, this HSP threshold where you have number of residues aligned, percentage sequence identity. This curve here is the so-called HSP curve, and now plus 5 is this, plus 10, plus 15, plus 20, minus 5, minus blah, blah. So below the curve, above the curve. And that's essentially here the value that you see. You see, if I use that to infer structure, you would have an extremely sharp transition, like a phase transition. This is just the same for both. If you look at localization, you still sort of have this S-shaped kind of curve. It's not quite going down to 33% because of some technical reasons. But this is exactly what I meant before. You're certainly not going to pick this to make a decision. Now the question is, do you put this one here or that one? And that ultimately depends on what is your tolerance for how much error do you tolerate. So as you see, if you put your threshold into this point here, you will be 90% of the decisions right. If you put your error here, you will be almost always right. So it depends on what you want to get out from the tool. What do you believe is the price of going further down or keeping the threshold higher? What do you pay for that? Or what is the... Why not always pick the higher threshold? What could be the problem with that? Yeah?
You might have very few
hits. Exactly. You may have very few hits. So if you want to have a higher probability that you have any homology-based inference to make, you go further down. And what will decide how far down you can get? Ultimately. So say, for the sake of the argument here, maybe this curve actually goes down and I just wiped out something underneath here. Maybe the curve actually hits the 33 somewhere here. Say it went to the 33. Would that be an okay threshold? So I would argue it's not a right threshold. Why not? Somebody else then, Stanislav.
Because we only have 3 classes and it's like doing random prediction.
Exactly. So let's follow up what Fados says. Where do you believe the threshold should be? So not at random, that's clear, because then if you pick the point that is as good as random, you can as well take a coin. Well, if it's not below random, you can say it's a sheep coin, so you can still do it, okay. But that is sort of a nonsense argument. But what could give you another reason to put a threshold somewhere higher? Let's involve other people. Anybody else? What could... Random is one way to define the lower end. What else could be a way to define the lower end, or to say something that you want to beat? A simple prediction method would be something else that you... And so essentially now we're turning full circle. Because later in the lecture today, I'm going to show you a prediction method. And I'm going to say that the prediction method has to be this one here, and this one has to be the prediction method. Essentially, these are two sides of the same coin. Anyway, so we can pick different thresholds here. What you also see is that using the HSSP curve as a way is slightly better than this one. Well, it is better. It looks better here. There's something else to be said that I don't have here. Now let's talk about the next one here. So sequence similarity, we have some idea of a threshold. Let's talk about text analysis, and that is work from Rajesh Nair here. And essentially Rajesh's idea was, what if somebody gave me this record here without that particular annotation? Could I take this description of a protein function and say, if the protein has that description, then it must be nuclear. Okay, so anybody in the room who can already say that for that particular protein it's clear that it will be nuclear? Has anybody in the room... So if you have had enough exposure to biology, then you will at some point be able to say that. There's something that gives it away here, yes?
DNA replication maybe?
Yes. So DNA replication, again we briefly discussed this on Tuesday. The nucleus is the place where you keep all the DNA, but it's also the place where you process all the DNA. So DNA replication is something that you do to DNA, and that is done in the nucleus. So if that wants to do it in the nucleus, at that point it has to be the nucleus. Okay? Here's another part, that's the transcription activator. It binds DNA. So there's sort of a bunch of things that indicate that this is a nuclear protein for an expert. Now, and there are other things like that. A chromatin regulator is nuclear. A blood coagulation protein. So when you hurt yourself, the wound is coagulating. And this again is done by the proteins that are sent outside of the cell. This is, now we have a multicellular organism, this is a slightly different story there. But again, these are proteins that float around in your blood, they lead to a coagulation. And if you have a blood coagulation factor, a particular protein, you know it's extracellular, because they swim outside in the blood, outside of the cell. Now, this is the kind of thing that experts know. But the question is, can we use these keywords in a way to recover the expert knowledge? And can we possibly do more than that? Can we sort of find non-trivial relations by a machine Now, in the machine, what I mean here is not machine learning. So, in a much simpler way, what could we do? We begin with a data set of proteins for which I know the subset localization. And then somehow we have to extract the keywords, because we want to read all of these keywords here and assign something. How could we do this? Again, we take a protein, shown here as a row. In the columns you see the keywords. So this is one particular keyword, although made up of two words, cell adhesion. And the 0 means it is in the description of that particular protein not to be found. 1, it's to be found, and so forth and so on, right? So for every single keyword that you have not cut out because it's too rare, or because it's meaningless, because it's in too many subset localizations, for all the remaining keywords, you essentially build up these vectors. Okay? Now, when you come, that essentially builds your, let's say, your experimental vector database. When you come with a new protein, you essentially build, for the query, you build its keyword vector, and compare it to the vectors. Now, the way Rajesh does this comparison, he says, well, the point is, for your query, you do not know whether all the keywords really are there. Having all the keywords there means a high level of annotation, means that there's a lot of things known about the protein. What if you only knew some things about the protein? For instance, only that it binds DNA. Right? Not that it is there's a transcription factor and. How could you sort of still make these vectors match? And you could still make these vectors match by having subvectors. By seeing, well, you turn in a particular column here, you essentially for one particular protein, you turn its vectors that are 1s to 0s. Because some keywords could be absent. And if you match that one, you're still doing well. If you match one subvector that is created by a missing annotation, then you're still matching. And that is sort of the idea. Ultimately, you measure that by some simple Shannon entropy, simple information content, and then you, yes?
Sorry, I have a question. What if, for example, we have a protein and it doesn't have any keyword, yeah.
Then you cannot do it.
All zeros.
No, then you cannot do it.
Another one, and it's all 0, but you will probably not.
No, you cannot. You cannot assign.
Okay,
so this method only works. So the question really is, what could you do if you had some keywords? Could you then assign subset localization? If you're back to the situation where you have 0 keywords, you can do 0 with this method. So you can predict very well your performance. With 0 keywords, your performance is 0. I mean, it could be random, okay? But this, as a programmer, most likely you would say it cannot do. You could actually throw a coin. Yeah?
I'm just wondering how useful this method is, because if I don't even know where in the cell the protein occurs, do I have enough textual information about it?
So first of all, sometimes yes. Secondly, the method Rajesh, this was a relatively, he did not spend much time on doing this method. He got a prize for it, it was very successful, but ultimately originally he did that for himself because some annotations were simply missing. Because some annotator knew all of that and knew that the protein is nuclear, just did not write it down there. Okay, and Rajesh felt like, well, okay, I should have a machine that recovers these forgetful annotators. That was the original reason. And then we found out through that method that in fact in many cases, surprisingly often people know a lot about a protein and do not know these things because they are non-trivial relations. The DNA binding is just the counterexample or the simple example that is not the common theme. Okay. And I'll get back on a larger scale to an answer in a moment, hopefully, well, whatever moment means. So then you come with a query, you compare to the database, and then you get some performance, and I should have shown you the performance first. The performance shown here, precision versus recall, or accuracy versus coverage. Blue is essentially, you're asking, I pick the first one and I say, that's my prediction. Bless you. How good would that prediction be? That's what the blue line shows you. The red line shows you How well would you do if you simply put forward both the first best predictions, the two best predictions, right? Okay, so what you see first of all is that for some proteins here, ballpark 10%, you get an error rate that's in the ballpark of 90%. If you remember the homology-based inference I showed you before, this is sort of the same ballpark. So we can, for some proteins, reach a similar level of performance. So when we go down here, we find more proteins, but we go down in precision here. This curve ends at 80-something, 82%. Why do you believe it goes down?
The more, the lengthier the number of amino acids, the less likely it is we have.
Not the amino acids. We don't use amino acids here. We only use keywords.
Okay, the keywords, the less likely.
Exactly. It's back to what you said before. Essentially, the further we go toward that direction here, the more essentially we get very, very few keywords. Now, my question to you is a different one. If you saw that plot, if that would be your method, would you say, I give only the top to the user, or I give the first two? So, ask the question differently. The second is good, but is it better to keep it out, or is it better to give it? How would you answer that question? Any idea? So red is above blue. So you gain. Do you? And ultimately that's the point. So if we begin here, you see that you actually gain 0. Because if you only looked at how many of these red ones are right, essentially none of them. Because otherwise red would be above, right? So until this point somewhere here, and I should overlay the numbers of things you find at that point, but essentially until this point here, there's 0 gain, only loss. Because you're not adding anything correct. Or most of the things, the correct comes from the blue. You're not adding on top. And then it is unclear. So you have to compare and ask yourself, for whatever you add, are you actually beating sort of the worst thing that you can imagine, which is random? So you have to see the answer to that question is simply given by the random background model. Do you gain more than random? Not are you increasing performance, but is 1 out of 2 still better than 1 out of 1? That ultimately is the question we had last time. But in this particular case, again, you can easily, last time I asked you that question in terms of recall and precision for the enzymatic activity. Did we talk about that? Actually, no, I didn't. We didn't talk. Yes, in the introduction of function, we didn't talk about that, did we? Maybe not. Maybe I cut this out. Anyway, so the point is, if you ask, should I add the second hit? You have to ask, do you add information on top of what a random addition would be? If yes, then that would be a good idea. If yes, and that would be a good idea, then the only reason could be that you have a user who would be confused by getting 2 answers. That would be then the only reason not to do it. But in this particular case, in fact, that does not survive. So motifs is one thing, but here's another thing. What I just now had was a sort of sequence, text mining capability. Now I'm looking at sequence motifs. Sequence motifs, there are 2 types of sequence motifs. One is sequential. What I mean is, if that were the protein here, there's somewhere a set of residues that decides how this protein is going to be localized. Think about it like a zip code. You have a short address. If that address is recognized by another protein, it binds to that short address and shuttles your protein, brings your protein into a particular compartment. Then there are so-called conformational motifs, shown here, where if you look at the sequence alone, there is no motive. But if they fold up in three-dimensional structure, these two bits come together, and together on the three-dimensional structure, they form a motif. But you would have to know the three-dimensional structure in order to say that's a significant motif. Sequentially alone, it would not be significant. Okay, these two types of motifs exist, and ultimately, most of the transport that we know is regulated by simple motifs is regulated in this way. So you find short segments on the sequence that decide where the proteins go, and in particular, so there are different sort of pathways from the cytosol in exchange to the nucleus, mitochondria, plastids and peroxisome, and then there's sort of a major path here that goes ultimately out for secretion. And this one here is regulated by something that is called a signal peptide. So most proteins that are destined for secretion, for going out of the cell, are captured by something that is a signal peptide. Signal peptide is N-terminal, so it's the beginning of the protein. It covers sort of 15 to 30 residues. It is cleaved, cut off, when the protein goes through the membrane to get outside, in most cases at least. It exists in bacteria, eukaryotes, and archaea, slightly different, but in principle, all of that is true for all of them that I'm showing you. And it has a simple, what you may call architecture. What the authors, and I forgot to put the quote here, it comes in the next slide. The authors essentially called architecture here is there's an N-terminal region, there's a hydrophobic region, there's a region that goes before the cleavage site, and then there's the cleavage site. And this you can sort of recognize by a variety of methods. The N-terminal region has often positive charges. The H region here is over 6 residues long. It looks like a, yes?
I'm so sorry, it's not obvious. Previously you said that cleaved site and N-terminal is the same thing.
No, I'm sorry, that's a mistake. I said the wrong thing. There is an N-terminal signal. And then there's I'll show you, I believe on the next slide, this more visually. So if you think about the protein, this is the first residue, and this is residue 60 or something like that. Then this is the N region, then there's a hydrophobic region here, the H, and there's somewhere here's the cleavage site. Mm-hmm. So this whole thing up to the cleavage site, you would call the signal peptide. This is a motif, it's a part of the protein that's just cut off. All it's used for is to move the protein outside of the cell. Once the protein is at its future destination outside, these residues are cut off and will never be used again.
And on the cleavage site leaves the cell?
Only everything after that here, exactly. So the protein is obviously much longer, so whatever comes after that here, that leaves the cell. Okay?
So it cuts before leaving the cell, not after?
It cuts in during the insertion into the membrane, which is at the point of leaving, essentially at the gate. Mm-hmm. It cuts at the gate. Okay? And these are the regions I showed. So the H region here is over 6 residues hydrophobic. As the C region, just between the cleavage site and the hydrophobic region, is very polar, is not charged. And you see from this description already, this is the kind of thing for which you can build some sort of machine learning device. And even simple machine learning devices are the case. Here, the simplest thing at the cleavage site, what you have is a letter. The higher the letter, the higher the probability of this letter is describing this cleavage site here. And you see there are particular motifs or particular letters that are used more often. And here are somehow the background frequencies of a method that sort of uses initially some hidden Markov model here by the first author is Henrik Nielsen and Gunnar van Heijne and Sven Brunnack here. Essentially, these are sort of the different regions, the N-terminal region, the hydrophobic region, the cleavage region. And you can see all of these regions present, then you know it's a signal peptide. You use all of that, you find the cleavage site and you know where the signal is. That ultimately is the idea. And there are a bunch of methods that do it. Some used hidden neural networks. Then I will get back to that. Later ones do hidden Markov models, and I'll get back to another level in a moment. There are tons, so this is a very successful method, because about roughly 10 to 20% of the proteins are secreted, but it's particularly important because all the bacterial all the bacterial warfare against people has to do with secreted proteins that come out. So recognize them easily through a method like that is extremely important. This is why this was quoted very often. This is why tons of groups have sort of jumped on the bandwagon and produced methods like that. The latest, just published in March this year, the latest solution from the group of Henrik Nielsen here at the DTH in Lyngby, Copenhagen, Denmark, signal P5 uses obviously deep learning technology, and this is sort of an advanced, let's not go through that. There's LSTM backward and forward and some other tools to essentially predict the signal peptide. And that seems to be the best thing that is currently available. There are other things like signal peptides. There is, for instance, something that biologists call the transit peptide, which essentially is the same thing for chloroplast. Chloroplasts are cell types that exist only in plant cells. It's somehow similar. You cannot use the same method. It's just the same idea. You have something that you see on the sequence and you have to build a data set. So the most important challenge here was to build a data set, a clean data set. And then once the data set was there, everybody could train the machine learning devices on it. And that has actually happened. But again, so the data sets are smaller here. There's a targeting peptide. Again, it's very similar. It's just biologists like different words. All essentially are the same. This one is for the import into the mitochondria. There happens to be a method that sort of used them all three, but that did not survive in time. Okay, and here's another thing, another story, and that thing is subcellular localization. And the localization story is the following. You have a piece, a little part of the protein is recognized by a protein called importin or transportin, so they recognize this NLS, nuclear localization signal, okay? Then they put the protein through the nuclear pore. The pore essentially is a complex of proteins that puts a hole into the two layers of the nuclear membrane and puts these proteins, the transporting protein and the target protein that binds to the nuclear localization signal is bound by the transportin and put through that pore. And then sort of it does this thing here, and some proteins are exported again and they use the same signal. And just because biologists like words, it's called a nuclear export signal. There's a nuclear NLS, the import signal, and an export signal. But in fact, easily enough, they are often the same. Now, when we began many years ago to work on this, then we looked in a database and we found there's a database of motifs here. We found one particular signal, and that sort of matched to 96 nuclear proteins from 30 families roughly, and it had a precision of 90% and it had a recall of 3%. So precision of 90% means that when I use this signal, and 90% of the proteins for which that had this signal were actually nuclear. And of all the proteins that I knew were nuclear, 3% had that signal. So this is not very impressive. It could be that we cannot do better. But we felt we can do better, and the way we did it, ultimately, this is before you find a database, you have to create one, and that's what we did. We essentially read the literature, and essentially read for every single paper that people wrote, hundreds of papers we read and asked, what did the people annotate as the nuclear localization signal? In red are these nuclear localization signals annotated by people. So the first thing you immediately see is they have very different lengths. Red shows positively charged residues. So there are two amino acids that are positively charged. 2 in 20 is 1 in 10. You see much more red here than 1 in 10. That's another thing. So you see immediately there is an abundance of red. There's an abundance of positively charged residues. Here's another type of motifs, and in the most extreme one we see extreme cases such as these. Here, then you see sort of bipartite motifs. You have things where you have a bunch of positively charged residues, a bunch of positively charged residues separated by something. And they together form a motif. Now, what we did is we essentially built something that we called an in silico device, in silico mutagenesis. We essentially asked, first of all, in order to be a sort of proper nucleolocalization signal, it has to match to proteins in the nucleus. And it should not match to any other protein. Okay? Now, if somebody gives you, an experimentalist gives you a motif, and it does match to other proteins, one way of doing this is you just throw it out. If you go to the database gives you thousands of entries, you're not thinking twice, you throw it out. If you worked on this paper for half an hour, you're not going to first throw it out. You're going to try to do something else with it. You spent half an hour on this thing, right? You're not going to throw it away in 2 seconds. So what else could you do? So you have a stretch of sequence, and actually it does map in proteins that are not nuclear. What could you do to that stretch?
Maybe there are some motifs for proteins outside of nucleus, which, in combination of our initial motif, define whether this protein is clearly outside of nucleus, like another step of decision tree.
Let's say, Stanislav, you give the answer to 2020. This is 2000. In 2000 the world was simpler. There was good and bad. Bad because you could not just find many other databases where you had many other motifs you could compare yourself with. Good because, you know, many things were easier. So you cannot do that. What can you do? Something much simpler. So the good thing is, again, you don't need to look for other databases because they don't exist anyway. You don't make a mistake in not finding them. Yeah?
You overweight it.
What do you mean by that?
You basically artificially, I guess, double it or something like this.
No, wait a minute. You match to proteins that are not nuclear. What do you mean by double?
So, do you match it just in the non-nuclear?
No, so you have a protein that you match in nuclear proteins, but you also match it in non-nuclear proteins. So what would be your first suspicion why that is the case?
Oh, the first
Yeah?
suspicion.
Yeah?
Go ahead.
Okay. It might be that it's nuclear maybe, and
Yes.
just right now outside.
True. So that's true. So it could simply, the problem with that statement, Lena, is it doesn't help me for my method. This is certainly true what you say, but that is sort of end of the game, throw the paper away state. I want to have something else. I want to try something else before I go into throwing it away. What could I do? I have a sequence that matches in non-nuclear proteins and nuclear proteins. I suspect the experimentalists were just not careful enough. Let's put it like that. What could I try? Well, the idea is, so if it matches in other proteins, maybe it's just not specific enough. Maybe the experimentalist had some evidence that the KRQ here, KRQR, is in fact the localization motif. But maybe actually the S and the T should also have been part of the nuclear localization motif. So I take my nuclear proteins that have this motif. In this particular case, if I had KRQ here, and I would simply ask, because both of these nuclear proteins here have an RST there, what if I use the motif by adding the R? Do I still match non-nuclear proteins? If not, then in fact I redefine it. I say my motif is KRQR. If I still find non-nuclear, I see, what about the S? So how much do I have to add? Can I ever, in my set of nuclear proteins, is there anything in common between them that I can extend a little bit further, and once I do that extension, I get a nuclear localization signal that is completely specific to only nuclear proteins. So I still match all the nuclear proteins that I have. I don't reduce recall, but I completely change the precision by design. I design precision to be 100%, right? And if I cannot do that, I throw it away. So then that's the point at which I say, okay, we don't know whether there's a traveler protein, maybe they didn't do a good experiment, whatever it is, I don't know. But as long as I can do that, I should as well do that. Now assume that we would have the other way around problem. That we would have a nuclear localization signal, but I only find it in one particular nuclear protein. What would you do then? I find it in no non-nuclear protein? And I find it only in one nuclear protein. So somehow the few sample kind of problem. Yeah.
But how that is even a motif? Like motif is like.
Fair enough. Because somebody says this is relevant for bringing that particular protein. I can experimentally measure that this is relevant for getting the protein into the nucleus. They may not. Let's not discuss your point. So what would you do? You're right, Stanislav. Yes.
The opposite. You shorten.
Exactly. And this ultimately is what we did. We required that there are at least two different families of nuclear proteins that have a motif, and none outside, so that precision remains 100%. And we built a small data set that had 214 of these motifs driven by literature. By definition, precision was 100%. This one at that point had a recall of 43%. So this was a relatively short period of time, and it actually has has turned into one very well-cited paper, and you could sort of reach 1,300 nuclear proteins with that. Now, when we looked on a 3D-dimensional structure, where would our motifs typically fall? Then this is one of those motifs, the nuclear localization signal. You all are not biologists. Is anybody in the room who nevertheless notices something with this motif? I show two different molecules. Can anybody recognize that these are two different molecules? One is in sort of ochre color or brownish color, and one is sort of grayish color. The one in grayish color is called protein. What is the one in the brownish color? Can you say it a little bit louder? I believe you said it right.
DNA.
Yeah, that's DNA. Exactly, it's the DNA double helix. So, any suspicion what we see here?
Some sort of transcription.
You go ahead. Say the simplest statement first. Your brain made the first step. Speak it or say it. Anybody else? So this is the nuclear localization signal. What appears to be the case? Yeah? Yes, Stanislav?
No.
Yes.
Are we preparing an RNA from DNA or?
No. I mean, okay, this is the nuclear localization signal. And it is DNA. And this seems to be exactly binding DNA. Right? And that is the funny reality. So what happens is, in the first step, you have this nuclear localization signal, the protein binds to it, that makes you import it into the nucleus. Once you're in the nucleus, the importing or transporting the protein that binds to the nuclear, to the NLS, goes off. And the NLS sits there. And what does it do? It binds DNA. Because this is actually why it's imported into the nucleus. Then comes the exporting, and binds to the same nuclear localization signal again and exports it. It's a very, very simple mechanism. As soon as that motif is visible, it either binds DNA if in the nucleus, it's imported in the nucleus if outside, or if it, without binding DNA, sits in the nucleus, it's put outside again to have another cycle. It's a very, very simple mechanism through one simple motif. Now, there is this issue in principle that we believe, and that goes back to Günter Blobel, the late Günter Blobel, who died a few months ago, who essentially was the first to push this idea of proteins moving around through the cell through these zip code-like motifs. Now, what we do know is the kind of thing that I described to you, this subcellular localization, the nuclear localization signal, or the signal peptide. Those kind of things are the exception. They are not known for most proteins. How could that be? Well, one thing could be that we just have more to discover. Unfortunately, these slides were done, or most of that was done almost 20 years ago. We have 20 years more results, and we still, for most proteins, we don't know the signals. How could that be? Well, one is, of course, a lot remains to be discovered. Another thing is that there are other motifs out there. For instance, it could be that there are all of these conformational motifs, right? All of the things where we need to know the three-dimensional structure. Or there are other mechanisms. So can you think about another mechanism? And I believe